So should we give the presentation in the evening class?
No, not now. I think this is for the examination point of view, it is important. That's why I just wanted. So you can give afternoon, no problem. Afternoon, 10 minutes exam, right? 10, 15 minutes. So
Okay,
yesterday we have yesterday we have discussed about the electron transport chain, and we discussed about what is the goal of the electron transport chain, and then we have in details of the complex 1, complex 2, complex 3, and complex 4 and ATP synthase also explained. And before going to the part, let me once again briefly summarize such that if you are not clear about the concept, once again I will explain very not going through the much details. And we said that to understand the electron transport chain and oxidative phosphorylation, we need to understand the bioenergetics. We have already said that some basic principles of the bioenergetics. The bioenergetics mainly deals about the transformation of energy in the living organisms, and we discussed about well, there are three types of systems in thermodynamics. We said that isolated, closed, and open systems. In isolated systems, neither energy nor matter exchange takes place between the system and the surroundings, whereas the closed system, only heat exchange takes place, not the matter, whereas in open systems, both heat as well as the matter exchange takes place between system and the surroundings, and all the biological living organisms and cells are the open systems. Then we discussed about the first law of thermodynamics, second law of thermodynamics. Then we discussed about what is the free energy and the enthalpy, that is the heat content of the system, entropy, that is the randomness of the system. Then we discussed about uh, the relationship between the enthalpy, entropy, and free energy, that is G is equal to H Ts, and then the delta. So delta. So G is equal to H minus Ts. Therefore, delta G is equal to delta H minus T delta S. And here, then we discussed about the what makes a process spontaneous. That depends on the free energy change, and if the free energy change is nothing but the free energy of the products minus the free energy of the reactants, and if the free energy change is less than zero, then it is a spontaneous reaction. If it is greater than zero, if it is a non-spontaneous reaction. If it is zero, the reaction is in equilibrium. Then we discussed about the electron transport chain. In electron transport chain, it is the electron transport chain is the process in which the high energy molecule ATP is produced. We have already seen that all aerobic organisms consume oxygen and generate carbon dioxide, and during this process, the glucose undergoes oxidation, and so when the glucose is oxidized, the we get the carbon dioxide and water, and this reaction has two half reactions. So here, first the carbons in the glucose, the carbon atoms of the glucose are oxidized, and the electrons are released, and these electrons are trapped in the coenzymes like NADH and FADH2, and then these coenzymes further transfer those electrons to the molecular oxygen. As a result, the oxygen is reduced to water. So that's what takes place in the electron transport chain. So now we know that in a glycolysis and the TCA cycle, and when the glucose is oxidized to pyruvate, and then pyruvate further to acetyl-CoA, acetyl-CoA enters into the TCA cycle. In both, when the glucose is oxidized to pyruvate and when it enters into the TCA cycle, we get 10 NADH molecule, NADH2 molecules, and these NADH and FADH2 molecules are high energy molecules. In the sense, they have high energy electrons in their structure. Here you can see this is the NAD and this is NADH. It is reduced form, it is oxidized form, and this is the structure of the FAD, FMN and FAD, and this is the FADH2. So these are the high energy molecules, the high energy reducing equivalents. They have the high energy electrons, and these electrons are transferred to the molecular oxygen in the electron transport chain for the formation of ATP. And another important thing is that when when NADH is oxidized, the delta G is minus 52.6 kilocalories, and for FAD it is minus 43.4 kilocalories. In NAD, when the glucose is oxidized, both glycolysis and citric acid cycle, we have 10 NADH and 2 FADH2. So therefore, total energy is minus 613 kilocalories per mole. But that means when the glucose oxidation, the Gibbs free energy is only the 686. That means so 90% of the energy is conserved in the form of coenzymes when the glucose is oxidized. So in the electron transport chain, there are three important events that takes place. One is the high energy reducing equivalents like NADH and FADH2 undergoes oxidation to NAD plus and FAD. And during this oxidation, the electrons are liberated, and these electrons are transferred to the molecular oxygen through various redox centers present in the electron transport chain. During this process, the oxygen is reduced to water. That is the second step. And the third event is that when the electrons are transferred to the molecular oxygen from NADH and FADH2, the proton gradient is developed across the inner mitochondrial membrane, and this you can see here. So this is the inner mitochondrial membrane, and it has the many 4 electron transport chain complexes and also the ATP synthase. And when the NADH and FADH transfer their electrons to the molecular oxygen, this is the molecular oxygen where it is becoming water. During this process, the protons are translocated across the membrane. You can see here these protons are important for the formation of ATP. From ADP, when these protons are again translocated back to the matrix. So that means these three events, what exactly takes place in electron transport chain is the oxidation of NADH and FADH2, and during the electron transport chain, the oxygen is reduced to water, and during the electron transport chain, the protons are translocated across the inner mitochondrial membrane. As a result, the proton gradient is formed, and this proton gradient is utilized for the formation of ATP. So this is the overview of the electron transport chain, and we know that the goal of the electron transport chain is only to convert reducing equivalents like NADH and FADH2 to the high energy molecule ATP. And these NADH and FADH2 are produced in the glycolysis and TC cycle when the glucose is oxidized, and there are many enzymes are involved in the ETC, and during the electron transport, a pH gradient is developed, and this pH gradient is utilized for the ATP synthesis. And this electron transport chain takes place in the inner mitochondrial membrane in case of eukaryotes, whereas you can see here this is the inner mitochondrial membrane, and that's where the electron transport chain takes place. But in case of prokaryotes, it takes place in the plasma membrane. And now coming to this, as I said that this process requires many enzyme complexes, and they are present in the inner mitochondrial membrane, and the terminal electron acceptor is the molecular oxygen. As the electrons are moving from the NADH or FADH to the molecular oxygen, the energy is released, and this energy is utilized to transport protons from inside to the outside, that is the matrix, to the intermembrane space. So as a result, the proton gradient would be generated, and this proton gradient is essential for the for driving the ATP synthase, which converts ADP into ATP. and this is the formation of ATP by ATP synthase using the proton gradient is also known as the oxidative phosphorylation. And since this process takes place in the mitochondria, and we need to understand the details of the mitochondria, and see, mitochondria is the storehouse of energy, we know, and it has many many enzymes present, many enzymes or many molecules are reduced coenzymes present, and most of the aerobic reactions takes place in the mitochondria in the eukaryotic cell, and the citric acid cycle enzymes and the fatty acid oxidation enzymes, all these are present in the mitochondria. In addition to this, there are many other enzymes are also present in the mitochondria. And coming to this, the number of mitochondria present in the cell depends on the cellular energy requirement. You can see here for unicellular algae, it has only one mitochondrion, whereas the protozoan Chavus has only 500,000 mitochondria, whereas mammalian liver cell has 5,000. But when you see here that white mussel has very few mitochondria, this white mussel, alligator jaw mussel is the example for the white mussel. In the same way, red mussel tissue has many mitochondria. Example, the red mussel of the migratory birds. So since the alligator has the white muzzle, so alligator can snap their jaws with high speed and high intensity, but it cannot continue this motion beyond a few repetitions because this has white muzzle, and it has a less number of mitochondria to provide energy. Whereas the migratory birds, they need higher energy for their locomotor movements. That's why they have the higher mitochondria in their muscle. And so that means the number of mitochondria in the cell or the tissue is dictated by the energy demand of the cell or the tissue. So this is about coming to the mitochondria size. So we know the size of the mitochondria is 0.2 to 0.8 micrometers in diameter, and the length is 0.5 to 1.5, and it has the two membranes, inner membrane and the outer membrane. And this outer membrane, the outer membrane has some proteins. They are also known as the porins, and these porins forms a channels and allows the some molecules which are freely soluble in water. And whereas the inner membrane is highly impermeable to the charged molecules and also the large polar molecules. Only uncharged molecules such as water and gaseous molecule like oxygen, carbon dioxide only can enter through inner membrane, whereas the protons and other polar molecules and the charged species cannot enter into the through the inner membrane. That means outer membrane has a freely permeable, whereas the inner membrane is impermeable to very bigger and charged species. And so in order to transport these polar and charged species, inner membrane has various transport proteins. And in the mitochondria, the inner membrane is folded, highly folded. That those foldings are also called as a cristae. And the matrix, this is the matrix, and the matrix has many enzymes like pyridehydrogenase complex and the citric acid cycle enzymes, and the enzymes that are required for the fatty acid oxidation, and also the enzymes that are required for the mitochondrial DNA replication, mitochondrial DNA transcription, translation, all are present in the matrix. So this is about this one. Now coming to the all the complexes that are present in the mitochondria in the electron transport chain. As I already said, that there are many complexes that are involved in the electron transport chain process. These complexes are mainly the four, that is the complex 1, complex 2, 3, and 4. In addition to this, the ATP synthase is also known as the complex of 5. And all these complexes have many oxidation reduction centers, and by having the many cofactors like FAD, FMN, ubiquinone, iron sulfur clusters, cytochromes, copper proteins, and so on, and all these oxidation reduction centers during the process of the electron transfer, they undergo either oxidation reduction to facilitate the unidirectional electron transfer. And the so in electron transport chain, if you see here that the electrons are always are transferred from NADH and FADH2 to the molecular oxygen in a one direction. That is why it is unidirectional electron transfer. So this unidirectional electron transfer is facilitated or is possible because in electron transport chain, all the electron transport chain complex, that is complex 1, 2, 3, 4, all these complexes are arranged in such a way that they start with the negative redox potential to the positive redox potential. So that means complex 1, you can see here, this is the redox potential, that is the, so the redox potential is the tendency of the tendency to gain or lose electron. So here the complex 1 is more negative, whereas the complex 4 is more positive redox potential. So that means the complex 1 has more negative redox potential than the complex 2. Complex 3 has more negative redox potential than the complex 2, and in the same way complex 4 is more negative than the complex 3. So that means the more the negative, the complex can easily lose the electron, the more the positive, the complex can easily gain the electron. That facilitates the unidirectional electron transfer in the electron transport chain. So that means you can also see here that again in the same way. So this is the complex 1 and this is complex 2, 3, and 4. The complex 1 is more negative, similar to here, and the complex 4 is more positive. That's why the electron is transferred from complex 1 to complex 4. So during the electron transport process, at any point of the electron transport chain, there is no chance for the electrons going back to the previous complex. For example, from complex 2 to the complex 1, that doesn't happen, because all the complexes are arranged in such a way that they start from the negative redox potential to the positive redox potential. And you can also see their redox potentials here, redox potentials here. So here we have once again one point you need to remember here, that the electron transport chain is taking place between NADH and the oxygen. Oxygen is the terminal electron acceptor, and NADH and FADH2 are the electron high energy reducing equivalent. They are electron donor. So that means all these complexes, the redox potentials of all these complexes should be between the NADH and the redox potentials of NADH and oxygen. This you can clearly see in this table. The NADH, the redox potential of the NADH is minus 0.32, whereas the redox potential of the oxygen here is the plus 0.82. That means the NADH has more negative redox potential, whereas when oxygen undergoes reduction, it has a more positive redox potential. And you can see here, this is the complex 1, and this is the complex 2, and the complex 3, and the complex 4. Complex 1 is more negative than the complex 2, and the complex 2 is more negative than the complex 3. Complex 3 is more negative than the complex 4, and so on. So that means all these electron transport chain complexes are present in the electron transport chain from negative to the positive, that facilitates the unidirectional electron transfer. So unidirectional electron transfer, the more the negative, the complex can easily lose the electron. The more the positive, the complex can easily gain the electron from the previous complex. That facilitates the movement of electrons from complex 1 to 2, 2 to 3, 3 to 4, and so on. And so therefore the redox potentials of all these complexes are lying between the redox potential of NADH and the redox potential of the oxygen. This is a point you need to remember, that then only the unidirectional electron transfer takes place. So now coming to the as I already said that it has many many coenzymes, many complexes. In addition to all these four complexes, the electron transport chain also has the two movable electron carriers. So one is ubiquinone, second one is cytochrome C. As you can see here, in addition to all the four complexes, you can see in this diagram little more clear. So in addition to all these four complexes, we have two mobile electron carriers. These are the one is a ubiquinone, the second one is a cytochrome C. So ubiquinone is a lipid soluble molecule. It can picks up electrons from complex 1 and complex 2 and pass it to the complex 3. So in the same way, cytochrome C, it can picks up electron from the complex 3 and pass it on to the complex 4. So these two are the mobile electron carriers, mobile electron carriers in the electron transport chain. So that means in addition to four complexes, there are two mobile electron carriers. As I already said that in the electron transport chain, finally the oxygen undergoes reduction, tetravalent reduction to water, and during this electron transport process, the energy is released, and the much of this energy is utilized to drive the protons across the membrane. As a result, the proton motive force is developed. That proton motive force drives the ATP synthesis. You can see here that complex 1, complex 3 and complex 4, all these three complexes are involved in the translocating of protons from matrix to the intermembrane space during the electron transport chain process. And whereas complex 2 is not directly involved in the proton of in expelling the protons, because during the electron transport process, the energy that is liberated from the complex 2 during the electron transfer is not sufficient to push the to transport the protons across the membrane. As I already said, that this proton gradient is very important, because when these protons are again coming back to the matrix from the intermembrane space, then only the ADP would be converted into the ATP by ATP synthase. So this is the process that takes place. You can also see here all these four complexes: complex 1, complex 2, and complex 3, and complex 4. All these complexes are composed of many subunits, and they have many cofactors. I just wanted to highlight very few here. Later we will explain in detail. Complex 1 has FMN as a cofactor. It also has iron sulfur clusters. And complex 2 is a succinated reductase, and it is also the part of the TCA cycle enzyme. It has the FAD as a cofactor. It also has the iron sulfur clusters. Complex 3, it is cytochrome C reductase. Again, it has many heme proteins, heme BL, heme BH, and cytochrome C1 and iron sulfur clusters. And the cytochrome C oxidase, that is the complex 4, it has heme A, heme A3, copper A and copper B oxidation reduction centers present in the electron transport chain. So that means when NADH is donating electrons to finally the molecular oxygen, the electrons are moving from the various oxidation reduction centers present in the electron transport chain. You can see here that during the electron transfer from NADH or FADH to various redox centers that are present in all these complexes undergoes sequentially oxidation reduction and so on. Finally, the oxygen molecule would be reduced to water. So that's what takes place in the electron transport chain. You can also see here that all these complexes are arranged in such a way that they start from the negative redox potential to the positive redox potential that drives the unidirectional electron transfer in the electron transport chain. And so this is about the overview of the electron transport chain. Now coming to the cofactors, as I already said that there are many cofactors are involved in the electron transport chain, as I listed just now, the FMN, FAD, and FMNH2, FADH2. These FMNH2, FADH2 can donate electrons one at a time. This I think this we will say later. So the various cofactors are the FMN, FAD, iron sulfur clusters, and the the iron sulfur clusters and the ubiquinone. And as I already said that it is a lipid soluble mobile electron carrier, then the cytochrome C. All these are present in the various in the electron transport chain, and to facilitate the transfer of electrons from the reducing equivalents to the molecular oxygen. And I just wanted to highlight here a few points that the in electron transport chain, the FMN when FMN and FAD is reduced by complex 1 and complex 2, the FMNH2 and FADH2 are formed. These FMNH2 and FADH2 donate 1 electron at a time, and within the same way iron sulfur clusters also can accept 1 electron and also can donate only 1 electron, and in the same way ubiquinone also can accept and donate only 1 electron. And finally the cytochrome C, it is also mobile carrier, it can also accept to carry 1 electron at a time. So only at the beginning, only FMN FAD can accept 2 electrons at a time in the form of hydrogen and becomes FMNH2 and FADH2, but they cannot they can release only 1 electron at a time to the subsequent electron carriers. And of course this is the structure of the how the iron sulfur clusters are present. You can see here these are the two different types of iron sulfur clusters, Fe2S2 and Fe4S4. In all these cases, you can see here the iron atom is bonded to 4 sulfur. The iron atom is bonded to 4 sulfur atoms. 2 are inorganic sulfur, 2 are the side chains of the cysteine. And in all these cases, in all iron sulfur clusters, during the electron transport chain, the iron undergoes oxidation or reduction depending upon the condition. So it switches between the 2 oxidation states, that is the Fe+3 to Fe+2 or Fe+2 to Fe+3 during oxidation reduction process. And in addition to this, the cytochromes are also present, and the cytochrome also can accept and release 1 electron at a time. The cytochrome, the heme A and heme B, and all the cytochromes are also present in the in the electron transport chain complexes. You can see here this is the ubiquinone. It is only to make you a little clear, as I already said that ubiquinone is a 2 electron carrier, but it releases 1 electron at a time. You can see here this is the ubiquinone, that is the oxidized form of ubiquinone. When it accepts 1 electron, it becomes a semiquinone. When it accepts another electron and the 2 protons, it becomes a ubiquinol. So that is reduced form of the ubiquinol. But this reduced form of ubiquinol, again, when it is transferring electrons to the next carrier, it can only again release 1 electron at a time and forming the semiquinone and again forming the oxidized ubiquinone. So when I say ubiquinone or coenzyme Q, both are the same. Please keep this point in mind. Then now coming to the details of various all these complexes, that is a complex 1 and complex 2 and complex 3, and we know that all these complexes contain many cofactors, and so we will discuss in detail about all these complexes. Complex 1, as I already said, that it is also known as the NADH ubiquinone oxidoreductase. So NADH ubiquinone oxidoreductase, it catalyzes the transfer of 2 electrons from NADH2 to the ubiquinone, NADH2 to the ubiquinone, that you can see here. So this is the complex 2 and complex 1, and this is the structure of the complex 1, and here it is NADH, and it transfers these electrons. Complex 1 picks up electrons from the NADH and donates to the ubiquinone, and ubiquinone becomes ubiquinol. So that's what that takes place in the complex 1, and it is a very complicated enzyme, and the structure is not fully elucidated. And in case of prokaryotes, the complex 1 has 4 different chains, whereas in case of eukaryotes, there are 14 homologous subunits are present. In addition to this, 2 to 20 to 32 additional subunits are also present. The additional subunits stabilizes the complex present and also prevents the electron leakage during the electron transport chain process. And this complex 1 has this complex 1 has many cofactors, as I already said that it has the FMN. FMN is the cofactor. In addition to this, it has several 8 to 9 iron sulfur clusters also present in this. And this you can also clearly see here. It has FMN cofactor, iron sulfur clusters, and also iron sulfur proteins, and the binding site for the ubiquinone. And so during this, of course, the structure of this enzyme is L shape. You can see here it has L-shaped structure, and it has the membrane bound compartment, and it consists of a multiple subunits, and it has the proton transporting activity. And the larger subunit, this is the larger subunit, it is projecting into the matrix side. It has the terminal NADH dehydrogenase activity, and also the FMN cofactor, and the connector. This is the connector. The connector is composed of the multiple subunits of the iron sulfur clusters. So this is the structure of the NADH. Now, if you see here that what exactly happens during the electron transport process. First, the NADH donate 2 electrons in the form of hydride ion to the complex 1, and these 2 electrons are passed to the FMN, and the FMN is reduced to FMNH2, and this FMNH2 is further oxidized back to the FMN and releasing the electrons to the iron sulfur clusters, and these iron sulfur clusters further transfers these electron to 1 electron at a time to the ubiquinone, and upon accepting the 2 electrons from iron sulfur clusters, ubiquinone becomes ubiquinol. So that means in this process, you can carefully see here that what exactly happens at complex 1 is that NADH, the high reducing equivalent NADH undergoes oxidation to NAD+, whereas the ubiquinone undergoes reduction to the ubiquinol. That is the net reaction that takes place at the complex 1. And during this process, of course, it is simplified, the same diagram in a simplified form. During this process, 4 protons are translocated across the membrane and across the membrane. So as I already said, that the exact mechanism how this proton pumping is taking place from the matrix to the intermembrane space when the electrons are transferred from NADH to the ubiquinone in the complex 1 is not very clear. That means the exact mechanism of the proton pumping is not clearly known, but the 4 protons are translocated from the matrix to the intermembrane space at the complex 1. So this is about the complex 1, and coming to the complex 2. So complex 2 is it is again it is, as I already said, that it is the integral part of the TCA cycle, that is the succinate dehydrogenase. It is also called as succinate ubiquinone reductase, and this complex 2 accepts electrons from succinate when it catalyzes the reduction of the ubiquinone to the ubiquinol, because so this complex 2 has the FAD as a cofactor. So this you can see once again clear. This is the complex 2. It has the FAD cofactor, whereas complex 1 has FMN cofactor. So during the reduction, FAD becomes FADH2, and this FADH2. So now this complex 2 has 3 identical multi-subunit enzymes, and each enzyme has again 2 subunits forming the head group, and also the 1 to 2 subunits forming the, you can also see 1 to 2 subunits forming the membrane bound stack. So this is the membrane bound stack, this is the head group, and but the head group has FAD as a cofactor, and it has 3 iron sulfur clusters, and also the membrane bound stack has the heme B and the binding site for the ubiquinone. And you can see here that as I already said, that the complex 2 is the integral part of the TCA cycle enzyme. What you are seeing here is this: when the succinate is converted to the fumarate, FADH is formed. So this FADH is directly transfer the electrons to the coenzyme Q, coenzyme Q. You can just see here the coenzyme, sorry, iron sulfur cluster. This iron sulfur cluster further in turn transfer the electrons to the ubiquinone. As a result, ubiquinone becomes ubiquinol. So this is the reaction that takes place in the at the complex 2. So the net reaction here is that the succinate, when it is getting converted into the fumarate, so FAD is getting converted to FADH2. This FADH2 is nothing but the FADH2. FADH2 is in turn donates its electrons to the iron sulfur clusters. Finally, to the ubiquinone becomes a ubiquinol. So that means in complex 1 and also in complex 2, the electrons are transferred to the ubiquinone. As a result, ubiquinone becomes ubiquinol. So ubiquinone becomes a ubiquinol. And this is about the complex 2. Coming to the complex 3. Complex 3 is also known as the cytochrome BC complex, or it is also known as the cytochrome oxidoreductase. And this enzyme catalyzes the oxidation of QH2 and the reduction of cytochrome C. You can just see here that now the electrons that are coming from the complex 1 and complex 2 are with ubiquinone in the form of QH2 ubiquinol. And this ubiquinol, the complex 3, the role of the complex 3 is only to transfer electrons from the ubiquinol to the cytochrome C, which is a mobile electron carrier. So mobile electron carrier. So this complex 3, if you look at the structure of this complex 3 transfers these electrons from ubiquinol to the cytochrome C by a special process known as the Q cycle. This we will explain later. And this complex 3 contains 2 copies of the same enzyme, and each functional enzyme has 3 main subunits. One is cytochrome C1, second one is the cytochrome B, third one is the risky iron sulfur protein. And the cytochrome C1 has the 1 heme center, whereas the cytochrome B has the 2 heme groups, and the iron sulfur protein has the 2 iron sulfur clusters. So this is the this one. And if you see that how this mechanism takes place, as I already said that the complex 3, the main function is only to transfer electrons from the ubiquinol to the cytochrome C. Ubiquinol is also mobile carrier, and cytochrome C is also mobile carrier, but the complex 3 is transferring electrons from ubiquinol to the cytochrome C. So therefore, this complex 3, if you see here, this complex 3, as I already said that it has 3 subunits, and it has cytochrome C, cytochrome B, iron sulfur, many iron sulfur clusters. So here, as I said that the ubiquinol, this ubiquinol transfer electrons to the complex 3, which in turn transfer those electrons to the cytochrome C. And the transfer of electrons from the ubiquinol to the cytochrome C by the complex 3 by a special process is known as the Q cycle. So in this, what happens here is that this complex 3 has the binding sites for the ubiquinol. You can see here ubiquinone or the ubiquinol, and so it has the binding sites at the intramembrane side and also the matrix side. And in the matrix side, it is known as the QN site. In the intramembrane side, it is known as the QP site or the QO site. And so this N side, it is known as the QN or QI site. So initially, when the QH2 at the beginning, QH2 binds to the Q0 site or the QP site in the cytochrome cytochrome B subunit, and as a result, the QH2 undergoes oxidation to the semiquinone. That means the ubiquinol, when it undergoes oxidation, 1 electron oxidation, it becomes a semiquinone, and this electron is transferred to the iron sulfur cluster in the ISP subunit, and this electron is further transferred to the cytochrome C, that is in the cytochrome C, cytochrome C1. and the cytochrome C1 further transfers this electron to the cytochrome C, that is the mobile electron carrier. So that means when the ubiquinol undergoes oxidation, one electron is carried to the cytochromes in one direction, and when it forms the semiquinone is further becomes ubiquinone by transferring the second electron to the cytochrome Bl and the cytochrome Bh, and then the cytochrome Bh. Now here this ubiquinone again moves into the in the lipid membrane, and it again comes and binds to the Qn site at the matrix side, and the electron, that is second electron, that is coming from the cytochrome Bl and the cytochrome Bh, again taken up by the ubiquinone, oxidized ubiquinone, and it becomes again semiquinone. So that means at the first half cycle, that means this reaction takes place in 2 half cycles. At the first half cycle, one ubiquinol is oxidized, one electron is transferred to the cytochrome, and the 2 protons are pumped out, and the one semiquinone is regenerated at the end of the first half cycle. In the second half cycle, another molecule of QH2 binds to the QP site. Again, same process takes place. The second electron is transferred to the cytochrome C, and the second one more electron is transferred to the cytochrome Bl and the cytochrome Bh. As a result, the semiquinone that is formed at the first step is reduced to the ubiquinone that is formed. Semiquinone that is formed in the first step is reduced to the ubiquinol by the another electron. So that means here in Q cycle, 2 ubiquinols or 2 molecules of ubiquinol QH2 are utilized, but one QH2 molecule is regenerated. Only one is used, but the 2 protons are translocated and the 2 electrons are transferred to the cytochrome C, that is mobile electron carriers. You can see here that totally the net gain is that only one ubiquinone is utilized, and the 2 cytochrome molecules are reduced, and the 4 protons are translocated across the membrane into the intermembrane space. So this I think in a step by step it is given in this. You can go through this one later, and so therefore the net reaction here is that 4 protons are translocated when the ubiquinol is oxidized in the electron transport chain, and 4 protons are translocated across the membrane from the complex 3, and so you can also see here 4 protons are translocated. Now coming to the complex of 4. Complex of 4 is a cytochrome C oxidase, and it is the cytochrome C oxidase is the terminal electron acceptor here, and terminal electron complex, and it has its main function is only to oxidize cytochrome C and also to reduce water. So this again you can clearly see in this diagram. This is a complex of 4, and its function is only to oxidize, to take electrons from the cytochrome C and donate it to the oxygen. So that means its function is to oxidize cytochrome C and reduce water. So during this process, again the translocation of protons takes place. For every 1 pair of electrons, 2 protons would be translocated. But when in order to convert oxygen to water molecule, it has to undergo 4 electron reduction. 4 electrons plus 4 H plus only, it becomes 2 water molecules. So that means for complete oxidation of one molecule of complete reduction of one molecule of oxygen, 4 electrons are required. Therefore the 4 H plus ions. Therefore during this process, 1 pair of electrons transport only 2 protons, and the complete oxidation of oxygen transport 4 protons. And if you see this structure of this enzyme, it has 2 functional units of the cytochrome C oxidase, and the same enzyme is present. Two enzymes are present, and each functional unit has the three subunits: subunit 1, 2, and 3. And the subunit 1 is embedded in the membrane, and it has 12 transmembrane alpha helices and the 3 redox centers, and whereas the three redox centers and the type A heme protein and also the copper atom, and the then subunit 2, this you can see clearly in this diagram. So this is the subunit 1, and subunit 1 has the 3 redox centers, that is the type A heme and the copper ion and A and type heme A and heme A3, and also the copper B. And subunit 2, so this is the subunit 2. It has again it has beta barrel domain, and this domain has the copper A site. You can see here, this has a copper A site, and subunit 3, it has 7 transmembrane helices and in the membrane, and it has no redox centers and the redox centers. And so if in the electron in the process, what exactly happens here is that the mobile electron carrier, so that is the cytochrome C, as I already said that it is a mobile electron carrier. It takes the electrons from the complex 3 and pass it on to the complex 4. So this is the mobile electron carrier. So initially the cytochrome C binds to the subunit 2. You can see here, this is subunit 2, and I think here it is not clearly shown. Subunit 2, and pass these electrons to the copper A site, and this copper A site can accept 1 electron at a time. As I already said that the ubiquinone, cytochrome C, copper, hemes, all these they can accept 1 electron at a time and also release 1 electron at a time. So and then this copper A further transfer these electrons to the heme A, and from heme A to the it is transferred to the copper B, and which is close to the heme A3, and the heme A3 is close to the copper B, and it forms a binuclear center, and that's where the reduction of the oxygen takes place. So when the electron is transferred to the when electron is moved to the binuclear center, the oxygen is reduced to water. And you can also see here that the one for complete oxidation of one molecule of oxygen needs the 4 electrons. Here 2 electrons required for the half O2 to become one water molecule. For complete oxidation of one molecule of oxygen needs the 4 electrons and also the 4 electrons. So therefore, when the 2 electrons are transferred from cytochrome C to the 2 electrons, in the sense the 2 electrons that are derived from the NADH, and when they are moving through the complex 2, complex 3, and complex through cytochrome C to the complex 4, the 2 electrons can cause the oxygen partial reduction of oxygen. That is the partial reduction in the sense half oxygen, half O2 is converted into the water, and complete reduction of one molecule of oxygen requires 4 electrons and the 4 protons. So therefore, for one pair of electrons, only the 2 protons are translocated across the membrane, that is from the intermembrane space to the from matrix to the intermembrane space. So therefore the conclusion is that complex 1 can translocate 4 protons, complex 2 locates 4 protons, and complex 4 translocates the 2 protons. Total 10 protons are translocated from the matrix to the intermembrane space when NADH is and when NADH donates its high energy electrons to the molecular oxygen in the electron transport chain. So this is what that happens. And last one is the ATP synthase. Once the electron transport once the proton gradient is generated, and this proton gradient is utilized to synthesize the high energy molecule ATP from ADP and by the ATP synthase. And this ATP synthase, you can see here, it is also known as the complex of 5, and its function is only to convert ADP and PA to ATP. And this complex has this ATP synthase has two subunits, and F0 subunit, F0 subunit, and as well as the F1 subunit. And the F1 subunit, it is also multi complex. F1 subunit has the enzyme catalytic activity, and each subunit has each complex has F1 and F0 complex has many subunits. You can see here, and here, when we see the structure of this enzyme is embedded in the membrane, and it has a characteristic knob and stack like a structure. You can see here it forms a, so the F1 subunit, you can see here this F1 subunit is composed of alpha 3, so 3 alpha subunits, beta 3, and 1 gamma subunit, 1 dou and 1 epsilon. Whereas F0 subunit, this is the F0 subunit, it is composed of A1 and the B2, and see here this B2, and also this 10 to 14 C subunit. This is the C subunits, and here the A, B, alpha 3 and alpha 3 and beta 3. So all these A, B, alpha 3 and beta 3 forms say a stator, and whereas the C and the gamma and epsilon subunits forms a rotor. So when the protons are when protons that are from the intermembrane space, when they are passing through this F0 subunit, that is between the A and the C subunit, the rotor rotates. That brings a conformational change in the stator, especially in the beta subunit, and the beta subunit has the ATP as activity, and this beta subunit converts ADP into PI. So therefore, as the protons are moving, and that brings a conformational change in the stator, and especially in the beta subunit, and it has the catalytic ATP as activity that converts the ADP and PI into the ATP. That's what takes place in the electron transport chain. And this, as I already said, that in the electron transport chain, as the protons are moving through the F0 subunit, the ATP synthesis takes place, but as we have already seen that there are 10 protons are transported when NADH is oxidized in the electron transport chain, and these 10 protons are, when they are moving through the ATP synthase, 3 ATP molecules are generated, whereas in case of FAD, it is 2 ATP molecules, but in new textbooks it is given 2.5, and the 1.5 we will tell you the reason why it is that. And we know that Peter Michel proposed the chemiosmotic hypothesis for the formation of ATP, because according to chemiosmotic hypothesis, during the electron transport chain, the proton gradient is generated, and this proton gradient is in turn coupled to the synthesis of ATP by ATP synthase, and because this proton gradient drives the ATP formation by ATP synthase, and since it is the proton gradient or the motive force, proton motive force is responsible. That's why it is known as the chemiosmotic coupling. So once the ATP is formed, and this ATP has to leave the matrix, because the ATP is also used in the cytosol for various metabolic processes. In order to transport the ATP, and the mitochondrial membrane has the ATP-ADP translocase, which is the antipoda. It translocates ATP and ADP in opposite direction. So for every one molecule of ATP comes out of the matrix, one ADP molecule enters, but ATP has 4 minus charge, whereas ADP has 3 minus charge. And when these molecules are moving in opposite direction, there is a net minus 1 charge loss from the matrix. So therefore, in order to compensate this, the one proton also enters into the matrix by the phosphate proton symporter. So that means when ATP molecule, for the formation of one ATP molecule, and 10 protons are required, and 10 protons are required, and in that that means one one NADH molecule, when it is oxidized, 10 protons are translocated. From the 10 protons, again, see 3 protons are essential for the formation of ATP, and the 1 proton is also utilized for the ATP translocation. So total of 4 protons are utilized for synthesis of one ATP as well as the exit of ATP from the mitochondria. And so this is about this. So now coming to the last, this is the P/O ratio. That is it is nothing but it is the number of molecules of ATP formed in the oxidative phosphorylation when 2 electrons are transported from NADH to the molecular oxygen. And still it is not very clear that we know that 10 protons are translocated during the electron transport chain when NADH is oxidized, and these 10 protons these 10 protons, as I already said, that 3 protons are essential for the formation of one ATP, and 1 proton is essential for the transporting the ATP out of the cell, because when the ATP is leaving the matrix and 1 proton is entering into the matrix from the cytoplasmic intermembrane space, therefore total 4 protons are utilized. Therefore, the 10 divided by 4, that is equal to 2.5. 2.5 ATP molecules are formed when the NADH is oxidized to the oxidized, and when the O2 is reduced. So this is the same. It is in case of FAD, it is only the 1.5, because FAD can only translocate 6 protons, because FAD enters into the electron transport chain at the complex 2. So therefore only 6 protons are translocated. As a result, the P/O ratio for the FAD is 1.5. So this is about the P/O ratio, and the many molecules in the electron transport process. These are known as the electron transport chain inhibitors. We have complex 1, complex 2, 3, and 4. You can see here the complex 1 is inhibited by retinone and pyridine and amytal. Complex 3 is inhibited by the antimycin, and complex 4 cytochrome C oxidase is inhibited by the cyanide, azide, and carbon monoxide. And then we have one more thing is uncoupling reagents. In uncouplers these are the lipid soluble molecules. They dissociate the proton gradient across the membrane. As a result, the electron transport chain continues, but without the ATP formation. One uncoupler is the 2,4-dinitrophenol. As you can see here, the 2,4-dinitrophenol, it takes the proton from matrix side, from the cytosolic side, and it releases the proton into the matrix side. As a result, the proton gradient that is generated during the electron transport chain would be disrupted. As a result, without the proton gradient, the ATP formation cannot take place. That means electron transport chain will continue, because the 2,4-dinitrophenol is a lipid soluble molecule. It can take protons from one side and release the proton on another side. Therefore, the proton gradient would be disrupted. As a result, the electron transport chain continues, but without ATP synthesis. And this is there is another physiologically uncoupling molecule, that is the UCP1. It also uncoupling protein. It is also uncoupling protein. It also dissipates the proton gradient. I think time is up. I will just explain only one more. So and the last one is the shuttle mechanisms. So these shuttle mechanisms, in the sense, we know that the NADH that is formed in the glycolysis cannot enter into the TCA, into the mitochondria, because mitochondrial inner membrane is impermeable to the NADH, because no NADH transporters are present. In order to facilitate the NADH that is present in the cytosol to participate in the electron transport chain, the cell uses various shuttle mechanisms. One is the glycerol phosphate shuttle. Second one is the malate aspartate shuttle. In the glycerol aspartate shuttle, it is carried out by the two enzymes. So one is cytosolic glycerol 3-phosphate dehydrogenase. Second one is the mitochondrial 3-phosphate mitochondrial glycerol 3-phosphate dehydrogenase. In cytosol glycerol 3-phosphate dehydrogenase, you can see here that when dihydroxystrone phosphate is converted into the glycerol 3-phosphate, the NADH that is synthesized in the cytosol is oxidized to NAD+. In this, the dihydroxysteroid phosphate is converted into the glycerol 3-phosphate, and this glycerol 3-phosphate again enters into the mitochondria, and where the mitochondrial this glycerol 3-phosphate dehydrogenase is again converted back to the dihydroxysteroid phosphate by the mitochondrial glycerol 3-phosphate dehydrogenase, because cytosolic glycerol 3-phosphate dehydrogenase uses NADH as a cofactor, whereas the mitochondrial glycerol 3-phosphate dehydrogenase uses FAD as a cofactor. Therefore, the NADH that is in the cytosol, in order to participate in the electron transport chain, it uses this shuttle. As a result, the cytosolic NADH becomes the mitochondrial FADH2. So this is the way the NADH that is produced in the cytosol in the glycolysis uses this shuttle to enter into the electron transport chain for the generation of ATP. And this you can also clearly see here that it's the same process you can see here. So second one is the malate aspartate shuttle. This malate aspartate shuttle again is catalyzed by the malate dehydrogenase. And you can see here when the oxaloacetate is converted into the malate, the NADH is oxidized to NAD+, and this malate enters into the alpha ketoglutarate malate carrier present in the mitochondrial membrane. This malate again further converted into the oxaloacetate and forming the again NADH. That means the NADH that is present in the cytosol is regenerated once it enters into the mitochondria, and therefore in malate aspartate shuttle, the cytosolic NADH becomes the mitochondrial NADH, whereas in the previous case, the cytosolic NADH became the mitochondrial FADH2. And of course, this oxaloacetate is further undergoes transamination to the alpha ketoglutarate, and this alpha ketoglutarate again comes back. Again, this cycle continues. That means here the NADH that is produced in the cytosol in the glycolysis is not entering directly into the mitochondria, because electron transport chain takes place in the inner mitochondrial membrane. So, but there is no transporter for the NADH. So, therefore, the cell uses the either the malate aspartate shuttle or the glycerol 3-phosphate shuttle, and this glycerol 3-phosphate shuttle is mainly active in the muscle and the brain, and therefore, and it is malate aspartate shuttle is mainly active in the liver, kidney, and heart. So these are the through shuttle systems are used to transport the NADH that is produced in the glycolysis to enter into the electron transport chain process. So this is about the electron transport chain and the oxidative phosphorylation, and it is very, very important even for the exam, and all of you should thoroughly go through this one. I think it is already time is up. Sorry, students, and this is the important. That's why I just covered.